Hello, everybody. Ah, okay. Did you survive the dinner? Are you awake? So, huh? I hope your synapses have worked this night and you still remember what I tell you yesterday. Um, in case you don't remember anything, I have this small resume. Um, so, we were trying to define biological neuronal networks in order to replicate electrophysiological data from the brain, like brain signals, and try to understand cognitive behavior and different tasks. And I've spent a lot of time talking about neuronal models. So this will be our standard model, the Hodgkin-Huxley, with 4 differential equations for the membrane potential of the neuron, and all the currents that are entering in the neuron, the synapses currents and the ionic currents, and different differential equations for the gating variables that can open and close. So this is X for M, H, and N here. And the typical situation is that a neuron receives external inputs and do a small excitatory postsynaptic potential, so increase a little bit, and then if the neuron receives a lot of external inputs, eventually it reaches a threshold and emits a spike, so the neuron fire. So we discussed how to simplify it or how to biologically improve the model. Then we did more or less the same for our synapses model. So now the synapse is a ionic, is a current that depends on neurotransmitters, but it's look like the ionic channels, and the current, synaptic current, will enter as a new term here in the equation, in the differential equation for the potential. So the synapses can also have gates that open and close with a specific rate, or can be just something that increase and decrease. But there is these conductance that are important values for our synapses, so our weights. So then we have neurons that are biologically plausible. We have weights, conductance, synapses that are biologically plausible. And mainly, they are changing in time. So we have a dynamical system changing. What was missing from yesterday is that we can also talk about synaptic plasticity here. So before we go to network topology, the third axis here, I will continue a little bit with synapses model, but thinking about how these synapses can change. So changes in synapses are related to learning and memory, and we can thinking about learning in artificial neural networks in training process, but we can think about learning in biological network in a sense that it's not separate. You are changing synaptic weights, learning and storage memory, everything in the same assembling of neurons, so in the same system, and just evolving time, the process is continuous in time for to have an idea. Then we can have different rules for these changes. They can be, in our case, will be very biologically inspired. So people did experiments, electrophysiological experiments, measure the excitatory postsynaptic potential, see differences in these potentials, and see that the synaptic weights change. So in nature, in biology, the synapses are changing. And these changes are related to learning process and memory process in very different experiments. I will mention some of them, and people block some neurotransmitters, and animals cannot learn or cannot remember. So I hope you all remember the class today. Everything is working in your brain, so you had a lot of synaptic plasticity during these nights. So the synaptic plasticity is the ability of the synapses to change conductance and weight in response to activity, to neuronal activity, or to experience, or to a task, or something that was external and If you receive a stimulus, then this stimulus make your neurons to fire, make more activity, and then these synapses change, and you have this plasticity. So it's considered the basic phenomenon for learning and memory, and typically we say that we have synaptic potentiation when there is a strengthening. So when the synapse increase, a G value goes to a G plus something. So this G here, this conductance, the facility of the ions or neurotransmitters to pass through the cell, it increases. Synaptic depression is the weakening, so when the G decreases for somehow in every step or with a specific rule. Then these follow the idea that have been repeated a few times here during the course, that neurons that fire together wire together. So if neurons repeatedly fire together, probably the synapses between them are getting stronger. So if you have neurons that respond to an apple or to a red circle, they will typically fire together. They will typically have synapses that increase the conductance. More recently, it has also been suggested the opposite. So if there is a failure that the neuron A fires, but the neuron B, for some reason, for inhibition or because they are not connected, the neuron B do not fire when neuron A fire. So if this repeatedly happen, the synapses between A and B will decrease in value. So there are, in beginning it was an idea, an hypothesis, but there are a lot of experiments showing the way it can happen. So if we have this change in G goes to G plus A, we can see differences in this height of the excitatory postsynaptic potential. So if for some reason it's happening plasticity here during this small time, this difference here will be smaller, and then this difference here will increase, and then will increase. So each excitatory potential will be greater because G is changing. That's the idea. If there is depression, the opposite would occur. So the same random input arrive, but the EPSP is do not have the same value. Okay? Do you get the idea? Potentiation and depression. I will talk about these names again. So we can have short-term plasticity. That is something that we can see with very few spikes. So if we have a presynaptic neuron and a postsynaptic neuron, and this weight is changing due to some kind of short-term plasticity, when the presynaptic neuron spikes, fires here, in the first spike, the EPSP is small. In the second, it's higher, what I was saying. So the EPSP increase in very few milliseconds, hundreds of milliseconds, and we can see this increase. So this is called short-term plasticity, and the G of this, or the weight of this synapse is changing. How much it changed could depend on the frequency of the stimulus. So if this neuron is spiking more slow, so if these spikes are more separate, and then we have small frequency, then the increase could be small, and then there is an optimal point here, and okay? So it depends also of the frequency of this presynaptic neuron. If we have depression instead of potentiation, after few spikes, the same stimulus will produce a EPSP that is smaller than the first ones, okay? So in this situation, we would have a depression. And this could be described by, now the conductance is a function of time, it's not just a fixed number. I mean, the current is a function of time and is a function of the conductance, but now also the conductance is a function of time and can change with some kind of rules. In this case, this specific case, we can add this probability to the G in the end increase or decrease depending on the time between the last spike arrive. So just to have an idea, now we have another differential equation, not just only for the open and closing of the channels, but also for the changes in the conductance. Okay? Sounds good? Questions? So the most common word in this part is long-term potentiation or long-term depression, so LTP or LTD. They are changes that remains after sometimes after a protocol. So you have a lot of stimulations here, and more or less they produce the same EPSP. Then you realize a procedure, you stimulate the cell in such a way that it was developed in an electrophysiological way in a dish, and they realize that after you do this procedure, you promote plasticity, you promote long-term potentiation, and the next spikes, they have higher EPSP. And more than that, this remains a lot of time. So this, in the end, like after many minutes here, you still have a EPSP that is larger than the EPSP before the protocol. So something happened here. You show something for your network to learn. You did something with your animal, or in the dish, you stimulate your cell, then you go to this change, and this change remains for a while, okay? So that's the idea. In the opposite case, there is a kind of protocol that promotes LTD, that promotes depression. So you have your APSP here, you do this stimulation with low frequency, and then after a while, you have that any stimulus similar to the ones that you were giving before, they will have EPSPs that are smaller. So the G will go to a G minus A. In this case, G will go to a G plus A. Okay, sounds good? When you read the papers as a physicist, at least it's kind of, oh my God, how this works. They do like a soup of neurons and mix everything and put a lot of things and then put in a culture and then stimulate. Thank you. I know. It's crazy. You mean still neurons? Like, it's still, can you compare to the brain? But yes. It's just because we are not used to, and fortunately, I don't have to do that in my computer. Oh, yeah. Excitatory postsynaptic hypotension. So postsynaptic hypotension is the effect of doing some kind of stimulant. I think I should put, more times in the slide, sorry. The effect, you mean here, this is minutes. How long it remains? Typically in these experiments with culture, this is 1 hour, not much than hours. But perhaps there are experiments that they try to see if it can stay for days. But mostly they are considered long if it's hour. So, but it can remain for hour. But the culture also goes to destroy. But the effect, in principle, could be like stable. For longer time? No. I mean, in the way they do these experiments, they just do for hours. But probably in our brain, like even if you learn something, if you consolidate a memory, so the strength of these neurons that are related to that will remain like higher than before you learn for forever? No, but I mean. More questions? I'm happy with questions. Especially these ones that I really have to think, okay, next time I will write again EPSP many times here because it's very important. And I know it's a lot of terms and we just, what? G? Conductance, please. G, conductance. Erbian rules and learning. Okay, neurons that fire together, wire together. Perhaps they could be related or not, but then a lot of different experiments happens to show that this is probably true. So these ideas of neuronal assemblies that are related to our perception, that are related to specific objects have been tested in very different experiments. And especially, I like this idea that LTP was necessary the formation of memories in vitro. So animals that NMDA receptors were blocked, so people goes and block the NMDA by pharmaco stimulus, and now they could not remember where is the they could stand without swimming, like the platform. They cannot leave the platform. They want to stop swimming, but they can't. So in vitro analysis showed that this LTP it was in fact blocked. So if they blocked NMDA, they blocked LTP, and the animals are not able anymore to learn how to get off the swimming pool. So they want to go out of the swimming pool, okay? And if they cannot learn the position, they get stressed, then this is not nice. So if you block NMDA, you avoid them to learn. So somehow LTP is related to learn. And genetically modified mice with NMDA receptors, they have poor spatial navigation capability. So they are unable to learn how to navigate in a new box or in a new maze, okay? So learning, memory, they are related. Typically, we looked for these things on the hippocampus of the rat, and there are many of experiments since many decades showing that probably at least these ideas make sense, okay? So perhaps someone have a new idea that is also compatible with all the experiments, but so far, this is the way we could explain the experimental results. And then, when I start my PhD, it was very fancy spike time independent plasticity ideas. So you can have long-term potentiation if neuron A fires before neuron B, and you have long-term depression if neuron B, the postsynaptic neuron, fires before A. And this was closely related to the time difference between the two spikes, the spikes of A and B. And many experiments in vitro have shown that this relation is valid. So this confirmed the idea of Herbian rule. Could be this, okay. The connection is unidirectional. If you have both connections, then one will strengthen, the other will weak. Okay. But there is a connection from A to B, isn't that? That's where, okay, with the connection, then B fires before A a lot, so there is a decrease. The opposite? No. Okay. They are the physical connection, the anatomical connection is really from A to B. So there is no way of an inversion of that. So chemical connections are just in one direction. Perhaps you go to 0, but you not go to negative value, so you do not invert. And then even if you go to 0, the anatomic thing is still there, but they are not releasing neurotransmitters anymore. Yes, I'm not aware of a specific experiment to see spike time independent plasticity in an autopsy. But for sure, if the neuron has an autopsy, so the firing rate of the neuron can alterate the conductance of this autopsy. I'm not sure if the rule will be exactly that. But yes, he's the presynaptic and the postsynaptic at the same time. But I mean, he's the same, but the connection goes from one region, like there is the soma and the connection is in another region. So it will be a difference of the spike, and then the synapses take a while to arrive in the other part of the neuron. So it's not instantaneous, so you will still have differences in time. So it was discovered by Markram, which is a huge name in neuroscience because the Blue Brain Project, perhaps you heard about in Lausanne, to simulate a huge amount of biological neural networks. And the idea is that we can have like a G when we have long-term potentiation, when we have the causal order, A first and B after, we have long-term potentiation, so G is equal to G plus something. So it's not just A, but it depends of how long the presynaptic neuron have spiked. So it depends on the spike timing difference between the spikes of the two neurons. So if it's pling, pling, very close, the increase will be larger than if it's pling, pling. Okay? So more or less, this is the idea. So if they say pling, 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 there is a big increase. If they say pling, 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 there is a smaller increase. And this is what our picture is saying here. But then you can define this as an additive or a multiplicative. You can say that the increase depends on the value of the conductance or not. Same, and both cases have more or less the same shape, so you can use both. And the idea of the opposite, the long-term depression, is the same if you invert the order. So pling pling is not a good example. It should be pla pling. But it's hard to say, so you understand. But then they realized that we can have a lot of different things. It's okay, the typical one is this, but then if we look to inhibitory neurons, the situation is completely different. Sometimes we have just long-term depression. Sometimes we have long-term potentiation if the interval is small, no matter the order, and long-term depression if the interval is large. So we can have exactly the opposite here. We have everything in real biological systems. So more or less, depending on where you are, hippocampus, neocortex, and the kind of neuron and the kind of synapses, you can see many things. And I also have, I don't think I have time here, but I also have a work saying that they typically talk about causal order in the sense that A first and B second, but we can still have a causal order with B firing before A if we have some kind of repetition oscillation. So if they are synchronized with a negative phase, we call that anticipated synchronization. And this could happen with if we have these inhibitory autopsies that I mentioned before. And then we would have long-term depression even in a causal situation. But perhaps in the next class. So finally, we can really simplify the Herbian rule to remember our artificial networks. So now we have the neurons, the signal function here, and then the weights will change as we saw in the blackboard a few times with a proportion of the activity of the neuron. And this kind of learning with, that depends on the firing rate is the most common in artificial neural networks, but can also describe biological neurons depending on the situation. Okay? Questions about synapses model, synaptic models or synaptic plasticity models or before? Feel free, I will go for water. If you are okay, now we have 2 of our 3 ingredients. We know how to model neurons, we know how to model synapses. Now we'd like to ask how to model topology. Okay, we can thinking about artificial networks. We can just say, okay, mathematicians, you know a lot of graph theory. There are plenty of different structures. Let's see what happened with all of them. I mean, every time someone approaches me and, oh, did you try the Abelis, or did you try Betty network, or no, I didn't try because there are a lot. So we can do, could be fun, but if you have an idea why to use, I would be happier. But we can do like an exercise. So we can put different neural models and different synapses in whatever network you want. And see what's going on and compare differences between these architectures. I would prefer to thinking about more biological neural networks that are similar to the brain. I hope I have time in the end to talk a little bit about complexity and entropy, and that's why I was asking Oliver. When we know a tool, we think to use this tool everywhere. But then, if the brain is not a very or totally regular network and is not a totally random network, we expect that if we measure the entropy of this network somehow, we would have something in between regular, that would be 0, and something smaller than 1, that would be if it was random. So something in the middle. But now we could expect that if we find a way to define complexity somehow, this measure of complexity should have small complexity for regular and random, and larger complexity for our brain network. Sounds like makes sense. It's a good fashion way to hands-on and to try to say that it would be nice to find this complexity measure, and we can think that the brain would be somewhere here and not the artificial ones, but each artificial network would be in another place, and each complex system that you can think about, social, traffic, airplanes, would be in a point of this plane. If we would like to do with brains, with real humans' brains, no? But whatever animal you want, the idea would be the same. We first need to measure the structural connectivity. So there is really synapses going from one region to the other. We have these cables conducting the electrical current, so we can define a matrix that really have an anatomical connectivity of the brain, and we can go in many details. We can parcellate the brain, I'll talk about that. But then we can define which kind of network we have from the real connection, the structural connectivity. Perhaps if you are a god that know everything, you can say, okay, that's the topology of the structural connectivity of the brain. It exists and is this one. We would have that at some point in our life. But then, even with this structural connectivity, we can have different functional connectivities. So even if you know exactly the topology of the structural connectivity, sometimes some synapses are activated, some synapses go to 0, so sometimes you are not using a pathway and you are using another way, and then you can define a different functional connectivity for the same structural connectivity, for the same brain, the same animal. They would have different functional connectivity during different experiments, during different cognitive tasks, or just in the same cognitive task in different days. So we are changing the way we are perform the same task. And our brain is changing, and we can do the same tasks using different ways, probably. So at least different matrix of correlation of dynamic activity. Then you have to define both of them in your model, or you have to define the structural one and see if it reproduces the functional one, or you decide to use already the functional one. It depends on what kind of question you are doing. So you have the problem of parcellation versus integration. That is, okay, you have your whole brain here, 10 to 11 neurons. You can do a network of 10 to 11 nodes, but then you can say, no, we should group these neurons and do a network with cortical regions. So visual area, auditory area, so far, But then we can say, okay, inside visual area, we have 7 other V1, V2, V3. Then you have to decide the scale that you are going, and you are trying to not lost information, so you want these regions to be integrated somehow, and then depending on these choices, you will have different networks. Typically, they have these atlas that are more used in the literature, more common, and then everybody use more or less the standard atlas that say, okay, we have a region that's called Broca region that is important for language. And then we have these visual areas with V1, V4 that are very important for this kind of visual experiments. So more or less, the academic community agree to use some kinds of atlas. But sometimes there is a new experiment showing that that part was doing something different, should be considered separate 2 parts, and then we always have different parcellations. So we can also think about the macroscopic scale in the sense that, okay, you decide we are going to use 34 parcellations here in one hemisphere, but then each parcel here, so each network here that is connected to another network here can be described by a lot of neurons, because in this very small region, we have a different network. So the way the macroscopic, the large scale is connected, can have a topology, and inside each node here, we can have a different topology among neurons. So especially in the cortex, we have these layers, things that sounds to be very important and appears in a lot of different experiments, that neurons behave different depending on the depth of the layer in your cortex. So we can go to, for example, the global connectivity of the macaque. We can see many regions of the brain of the monkey and see if they are connected. This is called sometimes the connectome of the animal. So which regions really are structurally connected, but then each region can be described by this small scale here. Or we can go, for example, to the hippocampus, and we don't have this layer thing here, but we have different regions, dentate gyro, CA1, CA3, and they are connected in a specific way that is very important for learning and memory and spatial navigation. But they are also receiving information from the cortex, and then the cortex have different layers that are connected in a specific way. So then if you want to study these, you will have this specific topology that is known, but then another group will say, no, these synapses here do not happen. And so it's more or less accepted how it is, but many of them have different experiments that show a little bit different. Then you have to test. It depends on the way the experiment was done, and it's hard to say for sure this is the connection between these areas. So there is this model from Potgny Disman that have worked with Roque a lot here in Brazil. So people from Brazil, Rodrigo, Renan, they work with this kind of network that try to really model each layer with real details of each layers. So they are 1 millimeter squared, and they have excitatory and inhibitory neurons in the proportion that they found in different populations. They have different density. I'm not sure we can see here, but the layers have different density of neurons and different connections interlayers. So there is this kind of model that is still, you can do in your computer, you can do the simulation not with all the different details of all different neurons, as some people in these huge projects are trying to do, but you put more or less homogeneous things in your neurons with very realistic network topology. So you lost in the diversity of neurons, but you gain in the reality of the topology. So it's always a choice, and this is a kind of thing that you can do. So, or we can go to, for example, C. elegans connectome that I mentioned yesterday. You have these 300 neurons. You know all the neurons, all the synapses. You know the direction. You have evolutionary information. So we know a lot about this animal. And the genome has been mapped, but still we could not reproduce a C. elegans in our computer. I don't know, like. But this, for people who are looking for simplified model, the most simple way of being processing information, I think it's a very nice model. So nowadays, we have many others on the internet. They are free. Thanks to these huge initiatives, we can go into the data, take a while to understand, but then we can have it and do our simulations or analyzing the data, whatever. I think it's open science. It's very important nowadays. Questions? I think for this, we are done. Like we have this recipe. Now I want to do a soup, or I want to do a biological neural network. I need a neural model, a synaptic model, a network model. Let's put together and let's have fun. Let's try to reproduce that data from patients with Alzheimer's, or let's try to reproduce data from mice. I don't know. Choose your preferred question in science and make it now. If you do not make, I will talk forever. I put a lot of things now that let's use. No? So this is what I do. Pedro, yes, I will show you here. No questions? Everybody knows how to implement a biological neural network now. So have you heard about this kind of thing? I will sit. If you have a cube, this is a Necker cube, sometimes you see the blue part in front, sometimes you see the red part in front. Do you know that? Like, have you paid attention of this problem before? If you just look to a cube, one of them, because I mark it just to make sure what's going on. And sometimes you see, you can try to choose, but sometimes it changes, your perception change. Can you do that with the paint one? I should put the no painted one, no? Let's see if I draw, if it works. Everybody's with me? Sometimes you change this perception. If I say, I will say first in Portuguese. If I say cajá, 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 sometimes you hear jaca, sometimes you hear cajá. In English it would be fly and life. If I say life, 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 sometimes you hear fly, sometimes you hear life. I don't know in Spanish, someone knows? Hablan español, algunos de ustedes, ¿no? ¿No hay un de este? No? Okay. In French, this would be plan and lamp. So for French people, if you hear plan, 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 sometimes you hear plan, sometimes you hear lamp. It's the same idea, but not with visual perception, but with auditory perception. So you can have the same stimuli, external world, reality, but then just because you are existing, just because of noise, just because you are an autonomous system, sometimes you see something, you perceive something, sometimes not. Okay, this could be just funny for family dinner, but we can also study that. And then this group in France, they did a magnetoencephalogram experiment with people hearing these plan and lamp conditions, and they measured the whole brain activity, and it's a group with 4 women. They realized that if you look to the oscillations of the activity in specific frequencies of the brain, so there are activity in 40 hertz, for example, and if you look to these oscillations, when the person reports that it's listening one word, is different than when the person reports that is listening the other word, and the difference in the oscillations are in the phase. So there is a phase difference between the brain signal when you are listening cajá, fly, or lamp, and when you are listening jaca, life, or plan, the other word, okay? And okay, we don't know if it's an epiphenomena, if they are related, but when you are listening in one thing, you have this oscillation in a specific frequency, and when you are listening the other thing, we have the oscillation with a phase difference. And this was not tested for the visual situation, but could be like when you are looking to the cube like that, when you are seeing the cube with this part in front, you would have one situation in your brain. When you are seeing this one, you have another situation. So if someone wants to do the experiment, I would be very happy to talk to. But then so far we have this experimental data. And then could we implement a biological neural network that have phase differences that change with the same statistical distribution that our change, like sometimes we hear cajá for a few seconds and then jaca, and then, yes, otherwise I was not showing here, but this is what we did. So let's use the recipe. Let's take an easy cavity neural model, just two differential equations, 500 of neurons here, 400 excitatory, 100 inhibitory, then another population that's very similar. Let's choose our preferred synaptic model here for AMPA and GABA. Let's put this synaptic current inside the neurons. Let's connect the neurons randomly because they are neighbors in these, because it's easier. We could do like layers of cortex, but okay, let's imagine that this is a cortical piece, a small population in the cortex that is connected to another small population in the cortex. So if they are oscillating, they could oscillate with a phase difference, and then perhaps we could find phase difference that change, that sometimes is cajá and sometimes is jaca. So we put external noise to make this activity continuous. We can all go to the more details, and then we connected unidirectionally these populations. So if just these neurons send synapses to this one, but not the opposite way, we can find different cortical regions in what this is happening. And if we say that the synaptic conductance here for excitatory between the populations and inhibitory inside the population are our important parameters, we can play with these parameters, and we can find, these are the students that did, Julio and Catiel, that did the simulations, and then we can find the activity of the first population in black and the activity of the second population in black, and depending on the values of our conductance here, we can have that the phase difference between the oscillations, so the difference of the peak of the black and the gray here in every cycle is changing. And if we plot this difference for each cycle, so we have, sometimes we have gray and black, gray and black, gray and black, gray and black, and then sometimes we have black and gray, black and gray. So sometimes we have negative values, sometimes we have positive values. I mean, sometimes black is happening before gray, sometimes black is happening after gray, or the opposite, as we define. But then we have a system that has a B-stability, that sometimes have this order of first population, then the second population, and sometimes jumps to another stable regime, so that has the opposite phase difference here. So we have, if we have a classifier to say this system is recognizing cajá here and jaca here. It's not, but at least the signal look like if we look to the distribution of this difference. We really have a big Gaussian here, so we have many moments in which we have this difference in the time peaks here that is negative. Sometimes we have this difference that is positive here. And then we can say that the system alternates from different situations that could be related to B-stable perception. So B-stability in phase in a very simple biological neural network could be somehow related to B-stability in perception. Okay? Oh. Relative to what? The density? Okay. So, I mean, in the real experiment with people, they do not report if they are confused. They just say, I mean, they press one button if they are listening Kaja and another one if they are listening Jaka. So you don't know how confident they are. So we don't know this information. So we just have that when they report that they are listening Kaja, there is one phase. And when they report Jaka, there is another phase. So we cannot compare to this intensity, at least the way they did. I don't know, like how could you imagine that they could be related to hallucination? Like, I mean, because hallucination, for me, it looked like you perceive something that is not there at all. And here, you can really perceive two things. But I don't know. The answer is I don't know if they are related somehow. I don't know the literature of hallucination, in fact. But it's a nice question. I mean, for me, the cube is there. It's not that you are creating something. It's only that you can really perceive something in a different way. But perhaps this is the same thing of hallucination. So we can look to the duration of each situation. So for how long you perceive Kaja, or for how long the phase difference is negative, and for how long you perceive the other one. And then you can do something like the number of occurrences as a function of the event size. And then this is more or less the number of cycles and this distribution similar to what is reported in behavioral experiments. So we see for few seconds one thing and then change to the other, and the distribution is not clearly following any kind of statistic here, but they are larger in the beginning. So far, it's a toy model, but this is the kind of thing we can play with. Questions? Everybody's hungry? Can I go for the last part, I promise? And I can sit like, let's take a brief. Okay, you can really do questions now because I'm not talking exactly about biological neural networks in the next slides. I will talk to a tool of data analysis that can be used to classify, but it's not a neural network. What I would like to advertise. You can ask Pedro in the following days because I have to leave today, but it's his work. And I think it's something that is more related to what many of you have asked me these days. So that's why I would like to show a little bit. Okay, so if you are interested in describe your systems in terms of statistical physics, you can think about measures of entropy. So you can think that if you have a paint that is very regular, this entropy will be 0. If you have a paint that is totally random, it will have an entropy 1 if you have a way to measure entropy of this kind of imagery. So, but everything else is in between. So no matter how beautiful is the paint, it's something just with intermediate entropy you expect. Then if you use this idea of try to measure somehow a complexity that is not totally related to the entropy, but have this property that regular and random would have small complexity, like it's not a complex system, a perfect crystal. It's not a complex system, a totally random signal, but many things in between could be considered complex systems, and you can study turbulences in these Van Gogh paints, and there are a lot of phenomena that you can think about that are related to statistical physics. So if you do the same with networks, we have this idea of the brain in the middle, and we can also do that with time series analysis. So if you have a brain signal or whatever signal you like and measure in your lab, or whatever image you have in your lab, if it's very regular, small entropy, if it's very random, high entropy. But then if you have alpha oscillations, brain signals here from EEG, electroencephalogram, or whatever, you should have something in between that. So we suggest a way to do this kind of analysis in graphs, or in time series, or in images, using the idea of symbolic information. So it's a symbolization technique proposed by Bonded-Pomp that says that if you have your time series here, you can zoom it here. So we have one point, another point, another point, 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 point. Or if you have an image, this would be the pixel of the image or the intensity every point in space. So if you have not a time series but a space series, it's also good. If you have power spectrum, or if you're having frequency or whatever, if you have a series, you can do the same analysis, which is define symbols. So if you decided to look just to 3 consecutive points, you have 6 possible symbols. You have increase, that's happening here, so the consecutive ones are increasing, and then the 2, 3, 4 here is also increasing. Can you see? So in purple here, we have this symbol. But then if we start in this point here, we have a small increase, then a small decrease, which would be this symbol here, 2, 3, 1. Are you with me? Can you recognize these symbols? So whatever you have as a series here, you can define to look for 3 consecutive points, and you can find these 6 points. Okay, you can also find one that is constant, but we will say that this is rare, not considered, or you decide to consider as a new symbol. It's just a matter of method. But then, now you can associate to this time series a probability distribution function of symbols. So you count how many times this symbol appears and come here and, oh, okay, 1, 2, 3, put 3 here. And then we count how many times this symbol appears here, 1, 2, 3, 4 times here, and then you mark here, okay? Are you following me? So then you have a PDF, a probability distribution function of these symbols. So now you can define an entropy of this PDF. Oh, ask me. Yes, exactly. Just to explain, for didactical reasons. In the end, I would say, okay, let's use more consecutive points. Then we have more possible symbols. Then we go just to 6 or 7, because if we go to more, it's computationally impossible. But this is a very good question. Okay, you can decide to use 4 symbols together here. Sorry, 4 consecutive points. Then now you have increase, increase, increase again, or increase, increase, decrease. So you have a factorial of the number of consecutive points you choose. Can you see that? Yes, there is this idea. If you have more points, more consecutive points, you have more information, so it's better. But then you have a computational limit. So what you do is see if it's stable. You try with 6 consecutive points and 7 consecutive points, and if your results are qualitative the same, so it's converged. You can test 3, 4, 5, 6. Or it depends on the number of how many points in your time series, because if you have, for example, 6 consecutive points, you have 720 options of symbols. So if your time series has just 100 points, you cannot look to a PDF with 700 possibilities, you know? The fluctuation will be large. And there is a compromise in how is your data, but there is no mathematical answer to say use 5. If someone finds this answer, please let me know. So with a PDF, so the probability of each symbol, you can calculate, for example, channel entropy. Channel entropy, as Boltzmann, almost P log of P, the summation here is just P log of P of this possibility plus P log of P of this possibility, and I was running. And then you can just have a number, so you just do a summation, put a minus in front, and you have a number, entropy, that characterizes this PDF, this distribution function, that characterizes this time series. So now, for each time series, you can find a value of entropy. Okay? For each image, you can find a value of entropy. Then you can decide to use whatever information theory function that you want. I mean, I could define that Fernanda measure will be P plus P squared. So I can just come here and calculate P plus P squared, and I will have a number, the number of the measure Fernanda. If this is useful, if this means something, probably not. But if you have a measure that makes some kind of sense, you can try to use different kinds of entropy, for example, here, okay? And then you can try to use, for example, the disequilibrium measure that we can call like a statistical complexity here, that it's more or less how different this distribution is from the equiprobability distribution in which all symbols have the same probability. If we define complexity like that, we have this behavior that we like, that complexity is small for regular and small for random, but can have larger values for intermediate values of entropy, just that. And then we did that. I can go in these details about downsampling, but I'm not going. You are all tired. Let's say that we can just define here a number for complexity and a number for entropy, so we can put in the plane complexity entropy here. For one specific time series, it will be in a place in a plane. For another time series, it would be in a different place of this plane. If we are happy, we can use this plane to classify our data. So lots of trials of visual tasks have points that are in these regions, and lots of trials of a different visual tasks are here. So you can use this plane to separate. Now, if you see a time series, you can calculate entropy complexity and say if it was recorded during a specific cognitive task or a different one. That's how we use it. I will skip the tau equal 1, 2, 3. If you want, I can go after. And then Pedro and Ícaro. So we analyze intracranial data from open data sets from patients that had epilepsy and went to the hospital and accepted to do these cognitive tasks in which they see a blank screen for a while, then they have a visual search task, then blank screen, visual search task. The visual search task, they have to say, what is the color of the square that it's, in this case, if the arrow is up, it's up to the star. So in this case, what's the color of the box that it's left, right? I don't know, this side of the star. And then you have just to do this visual search. And the patients are being recorded in different points here. So blue patients, red patients, green patients, they have these needles inside the brain. And during the blank screen, one very good needle here was measuring this activity. During this visual search, this one. And then for each trial here, Pedro calculated the entropy and complexity for different downsamples, and we found that there was a good one, but then there are these very beautiful regions that in every trial we can separate if the time series is from a blank screen or visual search areas. There are these brain regions that don't care at all for the, the task that you are doing, at least in terms of entropy and complexity, and you cannot say anything. And then we have regions that are more mixed. You can see some kind of difference, but this one is so beautiful. Then this is potentially useful for a lot of different things. You can think of groups of people with Alzheimer's and not. You can think of different sleep stages. You can think about different cognitive tasks, motor tasks, or different brain regions. So here you can compare during the same kind of trial. This region is more or less here, but this other region is more here, and different patients are in different places. So this one? Here, visual area. Yes, makes sense, but not all visual areas happen that, and other regions that are not visual happen that. Every time you can say, okay, but yes, you are right. Huh? It's close. It's close, and it's the same patient. So typically, if you are still close, you have some kind of correlation of the data. But then we have these other patients that is in the other hemisphere, but are similar regions, and it's here, and they more or less separate. So there is some kind of consistent, but it's very hard because we have just 5 patients with needles in very different regions, with very different, it's not like if you do an electroencephalogram, your data is very similar for each subject. In this intracranial data, it's very specific, very individual. Oh. I mean, each trial remains for 2 seconds in the screen. We did analysis from 100 milliseconds, 200 milliseconds, or the whole interval. There are small differences, but they are not abrupt. So, we didn't really test that, but looking, okay, these 3 points, they are more or less the same, and if you go here, they get worse. But this is not trivial with the other patient, the green one. So perhaps in the visual area, you have this distance relation, but also perhaps because in visual areas, you have defined subdivisions like V1, V2, and they can be in different places that are processing information like after the other, but okay, we have data from sleep that we have more patients and more things that we do this kind of analysis with all the brain, like, and you can see small of correlation with space. So there is an idea that things are happening different from prefrontal to the end.
Those are patients with epilepsy and the same type of epilepsy?
Not necessarily. But probably they are not having seizure, so they are not epileptic during the recorded time. A doctor just select the good data, but for sure you could say, okay, even without a seizure, the brain of an epileptic patient is different. Yes, but then I cannot open other brains. Okay? We can do more questions. This is our group. We are in Maceió. I should put pictures of the beach. This is a very nice place. Come here, come to visit us. And thank you. This is group. We need diversity. We need mothers in science. And it's in Portuguese, but for everybody, please spend 1 minute trying to think about a female or a non-white person or someone from a different region or someone that you would not just remember in the first moment just because this person is different from you, but perhaps this person can be very I don't know, an expert in the area as many others that you can remember. So when you have to invite people, when you have to collaborate, try to remember that diversity is important. Important since the very beginning, so I will not talk much about that, but kids in a very few age, they start to have gender stereotype, and so that's all. Thank you. Questions? Hodgkin Huxley? A resistor, yes, in the electrical circuit. Yes. There are a bunch of people trying to include quantum mechanics in these models. Not neuronal models, but smaller models, like in the release of neurotransmitters. So far, it's not well established. Considering electromagnetism, it's not necessary. I mean, you can reproduce the data with physics 3. You don't need Jackson and books, advanced books. I mean, if you already explain, it's because there is no important ingredient. Of course, if you want to see the volume conduction, so not in the neurons, but because, I mean, you can measure the magnetic field outside our brain. We use these machines. So it's not just going through the neurons, it's also going through the tissue. But then this is not important for the information that is being processed. Once it arrives to a neuron, it makes the neuron spike, and probably the information that we are using in our brain is from one neuron to the other. So what we do is we measure the magnetic field that this activity produces and try to infer this activity. But we don't care for this magnetic, I mean, this magnetic field outside is not passing information for my own brain. It's influencing, and it's a result of that, but perhaps we imagine so far, the community, that the information is going through synapses from one neuron to the other and not from in the muscles. I mean, I don't know. We imagine that neurons and synapses are the important conductance. Yes, there is a group in Unesp, I don't know, Rio Claro. I was in a jury, and they are doing Fokker-Planck equation and try to see that. But then you can do the math, but you are, so far, you are not comparing to an actual potential. It's not the same, like the actual potential is coming from the flux, and you don't need the Fokker-Planck equation. So it's just a different and very complicated way, and you want to arrive to the same result because there is what you measured. I mean, if you want to explain nature, you have to go to an experiment. So this is, but there are people trying to do that. But so far, the standard models are with physics 3. More questions? Everybody hungry? So feel free to write me if you want. I have to leave today, but Pedro is going to be here, and I'm trying to be accessible through email and social media. I don't know. Hope to see you again. Hope you enjoy the last days of the course. I'm so happy to be here these days. Thank you all.
a mesma coisa que você acredita, só que parecido. Não é a mesma coisa, claro.
Não,
É que eu,
podia ser a mesma coisa
eu
também,
trabalho
tá?
de
Vamos